Hello friends, good morning to all of you. This is Dr. Kanak Saxena, and in today's video lecture we are going to discuss about the genetic diversity analysis, which includes Metroglyph analysis and D-square statistic analysis. These both biometrical techniques, Metroglyph and D-square statistics, are widely used for the assessment of genetic variability in the breeding population. So knowledge of variability present in the gene pool or any germplasm is very essential to start any breeding program. So in this we are going to discuss about the what is variability, what are the different sources of variability, what are the features of polygenic trait, different types of polygenic variations, and different methods of assessment of variability. Variability is the presence of differences among the individuals of a plant population. In plant population, the differences present between the individuals is called variation or variability. Now, it occurs due to several reasons. Okay, due to the differences on the genetic constitution. Okay, or due to differences in the environment gene interactions, or essential for the resistance to biotic and some abiotic factor or some adaptability. The variations occur in the population between the individuals. Okay, now how variability occur, what are the sources of variability? It occurs either due to the spontaneous mutation, it occurs due to the recombinations, okay, or due to the natural crossing. Okay, the natural outcrossing, recombinations, and spontaneous mutations, these are the basic sources of variability. Now, after variability and its different sources, let's come to the features of polygenic trait. As we already know, in our previous video lecture we discussed about the difference between the quantitative traits and qualitative traits, polygenic traits. So these are the features of polygenic traits. They show the continuous variation and the small and undetectable effect of individual genes. So because several genes are involved, and many genes show small and undetectable effects. So there is no possibility of grouping into the distinct classes in polygenic traits. High effect of environment, they are based on the mean variance and covariance. Since they are based on the mean variance and covariance, there is a possibility of the metric measurements, and they are comparatively low stable because they are highly affected by the environment. Now let's talk about the different types of polygenic variations. There are three types of variations: phenotypic variation, genotypic, and environmental variations. Phenotypic variations are observable. Phenotypic variation include both genotype factor and environmental factor, and that's why the value of PCV is greater than the GCV, because PCV phenotype includes genotype and environment factor, while GCV include only genotype factor. Okay, the next is genotypic variation. These are the inherent capacity. It transfer from the parents to their offsprings, and such type of variation is does not affected by the environment, and it is measured as the genotypic variance. The third and last is the environmental variation. It is non-heritable, uncontrolled, because we can't control the environmental conditions. Okay, and it is measured as the error mean variance. Now this is the very important point, method of assessment of variability. There are four different methods of assessment of variability. The first is the simple measure of variability. It includes PCV, GCV, and ECV calculations. And next is the variance component analysis. It includes the various evaluation of progenies in replicated trial when grow in the definite fashion, like diallele, partial diallele, or line into tester. Such designs are used. The third is the Metroglyph analysis, in which Gilfer are drawn on the graph. D-square statistic, in which genetic diversity is analyzed by the cluster diagram. Now this is the first method of assessment of variability. It includes the range, standard deviation, variance, standard error, coefficient of variation. Now ANOVA provide the estimate of CV percentage. Simple measures of variability include PCV, GCV, and ECV. PCV is the under root of phenotypic variance divided by mean into 100, and the square root of variance is the standard deviation. Same for the GCV, it is the under root of genotypic variance divided by mean into 100, and for environmental coefficient of variation is the standard deviation of error divided by mean into 100. Now let's talk about the results or interpretation of the PCV, GCV and environmental coefficient of variation. If the value of GCV is greater than the PCV, then it means that there is a little influence of environment, and the selection in such condition would be rewarding. Okay, if PCV is greater than the GCV, then apparent influence of environment, because PCV includes the genotypic and environmental factor. Okay, so selection may be misleading because environmental factor is included. Okay, if the environmental coefficient variation is greater than PCV and GCV both, then it means the significant influence of environment, selection would be ineffective. In such a situation, in this situation, we cannot select any genotype. Usually, these two conditions are considered in the plant breeding program. Either GCV is greater than the PCV, or PCV sometimes greater than the GCV. Now, in this type of method of assessment of variability, various components are analyzed, in which the crossing of number of genotypes in a definite fashion, like line into tester, diallele or partial diallele, and the evaluation of the progeny in a replicated trial, then variance component are analyzed. Now let's come to the our third method of assessment of variability, that is Metroglyph analysis. This technique first time developed by the Anderson in 1957, when they are going to investigate several pattern of morphological variation in different crop species. Then it is a semi-graphic method, and it is based on the first order statistics. That's why it is highly robust compared to the d square statistics, which is based on the second order statistics. So results are statistically more reliable in this method. Okay, analysis is possible for both replicated as well as non-replicated data, and the pattern of variability is represented by the glyphs on the graph. Now what is glyph? Glyph are the small circle by which position of genotype or line is represented on the graph. Okay, so the mean values of one character for each genotype are plotted on the x axis, and the other character on the y axis, and each line occupies definite position on the graph, and such positions are known as glyph. The different lines may be represented by the different type of glyphs for easy identification, solid or open type glyphs, so that we can distinguish this is the exotic type of germplasm, and this is the indigenous line. Now Metroglyph analysis is based on three main steps. The first one is the plotting glyph on the graph. In this step, small circles representing the position of genotype on the graph is called glyph. Okay, and two characters having high variability are chosen. Okay, now one on the x-axis and other character on the y-axis based on their mean values. Here the mean values play important role. Okay, each glyph occupies a definite position on the graph. Okay, now based on the glyph, okay, divided into the different type of lines. The solid glyph represent the exotic or a hybrid type, while the open glyph represent the parents or indigenous line. Okay, after plotting the glyphs on the graph, the second is the depiction of variation. The remaining character out of two that shows the high variability, and we put on the x-axis and y-axis, the remaining characters are displayed on the glyph by rays. Okay, rays for same character have same position on the, and the length of the ray depends on the index value or index score. Now next step is construction of index score. How to construct? Now variation for each character is divided into the three groups: low, medium and high, with index score 1, 2 or 3 respectively. Sum of the index value is equals to worth of the genotype. Now the worth of genotype is calculated by adding all the index values of all the characters. Okay, by adding all the values of all the characters is equals to the worth of genotype. Now, the maximum and minimum score of an individual will be 3n and n, where n is the total number of characters included in the study. Now the last and final step of Metroglyph analysis is the analysis of variation, in which genotypes are divided into the various groups. I already said low, medium and high, and the maximum number of groups will be 9, and within and between groups variances are analyzed. How? Suppose there is a graph, as the one character show high variability based on the mean value, we represent on the x-axis, and the other character lie on the y-axis, and there is a low, medium and high. Okay, low, medium and high, three sections are there. Same here, low, medium and high. So there are, okay, 9 groups. Maximum number of groups would be 9. This. And the genotype for which are used as a parent in the hybridization program should be chosen from the different groups, and that represent the wide genetic variability. And this is how the variation is analyzed for the different characters within the group and in between the group. Okay, now let's better understand by using some worked example on Metroglyph analysis. Now let's better understand the Metroglyph analysis by using this example. Suppose there are 5 genotype, 1, 2, 3, 4, 5, and in which these solids A, B, C, these 3 are indigenous, and these 2 represent the exotic lines. Okay, now based on the mean values, these 2 character, N character and M character, that show the high variability, are put on the y-axis and x-axis. Okay, and all are categorized into the high, medium and low. Now these high, medium and low ranges gives information about the index score. The low range gives information about the index score 1, medium shows the index score 2, and the high range indicates the index score 3. So this is how Meteroglyph analysis is useful in different ways, like it helps in the study of the pattern of morphological variations in large number of germplasm lines at a time, and also useful for the classification of germplasm for various characters. Also the analysis is based on the mean value of the characters, so it can be applied to both unreplicated as well as replicated data. So this is all about the merits of the Metroglyph analysis. Now there are some disadvantage of this analysis also, that inclusion of large number of genotype, different type of, suppose 52 or suppose 150 genotypes are there, overlapping of glyphs on the graph is there, that's create the confusion and misleads the data for the analysis. So this is all about the Metroglyph analysis. Now let's talk about our second genetic diversity analysis, that is D-square statistic analysis. The concept of D-square statistics was originally developed by the P.C. Malonvis in 1928, and Rao in 1952 suggested this technique for genetic diversity assessment in plant breeding. Okay. D-square statistic method was widely used in the plant breeding and genetics for the study of the genetic divergence in various breeding materials, and this is the very important technique for measuring the genetic divergence, and it plays an important role because hybrid line and lines of diverse origin generally gives a greater heterosis compared to the lines which are related to the closely related parents. So this has been observed that in many crops genetic diversity arises due to the geographical separation or sometimes due to barriers of the crossability. So it plays very important role in genetic diversity analysis. So there are some important features of D-square statistics. It is a numerical approach compared to the Metroglyph. There was the semi-graphic method, and it estimates are based on the second order statistics, so less precision and less reliable, okay, compared to the Metroglyph analysis, which is the based on the first order statistics, so highly reliable and robust. And so the analysis is quite difficult compared to the Metroglyph analysis, and it is possible for the replicated data only. Okay, now the genetic diversity is represented by a cluster diagram. The analysis of d-square statistic consists of many steps. The first one is the selection of genotype. It means the germplasm line, strain, variety based on the phenotypic or geographical differences, and then the evaluation of the selected material in the replicated trials. Observations on each character are recorded. Now, biometrical technique variance for each character, covariance for their combinations are estimated. D-square analysis is based on the computation of D-square value and testing of significance. Now, in testing of significance, the significance value of D-square value is tested against the tabulated value of chi-square, where P is the degree of freedom, or P equals to the total number of characters under studied. So, if the calculated value of d square is higher than the chi-square tabulated value, then it is considered as significant, and vice versa. Okay, then the finding out the contribution of individual character towards the total divergence, then grouping of different genotypes into the various clusters, then estimation of average distance, intra-cluster, inter-cluster level, and after that construction of cluster diagram is done. So these are the major steps for the analysis of d-square statistics. Computation of d-square value, first of all testing with the chi-square tabulated value, then significant or non-significant. If significant, then finding out the contribution of each character towards the total divergence, then grouping of genotype into cluster, then intra-cluster or inter-cluster level, then construction of the complete cluster diagram. Now, in d-square analysis, a line diagram is constructed with the help of d-square values, which is known as cluster diagram. Now, this cluster diagram gives information about genetic diversity in an easily understandable manner. It gives information about the number of clusters, represent the number of groups in which a population can be classified based on the d-square values. Okay, now the distance between two clusters is measured the degree of diversification. Greater the distance, and between the two cluster, greater the divergence. Okay, and if the genotype is fall in this one cluster or in the same cluster or closely related cluster, then it considered as the less divergent genotype. So cluster diagram gives information about the relationship between various clusters. Now let's try to understand how to construct the cluster diagram by using some example. Suppose there are 20 genotypes and there are 5 characters, and these genotypes are classified into the 4 cluster based on the d-square values. Okay, these are the 4 cluster: cluster 1, 2, 3, 4, and the distance between these cluster is intercluster distance, and within the cluster we use intra-cluster term. Okay, these are intercluster between two cluster. Now, so the intra-cluster value of 1 is 2, for 2 cluster is 3, for third is 2, and for fourth is 1. These are the intra-cluster values, and the distance between two cluster, 1 with 2 is 4, 2 with 3 is 7, 2 with 4 is 6, 9, and 3 with 1 is 6, and 1 with 4 is 7. So we already know the square root of average intra and intercluster values. These square values are used for the construction of cluster diagram. If we want to construct, so arrange in these 4 columns, and the cluster 1 with 1, it means intra-cluster, the value is 2, 2 is square 4, here is 4. When we talk about cluster 1 with 2, 1 with 2, the distance is 4, so here is 16, 4 square. 1 with 3, 1 with 3. Okay, distance is 6, so d square value is 36. Here is 36. Now 1 with 4, the value is 7. 7 7 49. So 1 with 4 49. Okay, this is how we arrange the values during the construction of cluster diagram. Okay, now after the construction of this cluster diagram, we have to arrange the clusters and which one is the closest cluster and which is the farthest cluster. Here the cluster 1 is closest to the cluster 2. The minimum value is 16. Okay, and the farthest cluster is 49. And we know that the greater the distance between two cluster, greater is the divergence. If we have to select the parents on the basis of d square values, we have to choose clusters showing the maximum genetic distance and relative contribution of each character to the total divergence. So it is very helpful in the selection of genetically divergent parents for the exploitation of hybridization program. Now let's come to the merits of d-square statistics analysis. It helps to select the genetically divergent parents and it measures the degree of diversification. Maximum the distance between two clusters, more is the divergence, and determines the relative proportion of each component characters. It forces of differentiation measured at intra and inter cluster level, and also the most important, it evaluate large number of germplasm lines at a time. Now let's talk about the demerits of d-square statistics. This analysis is based on the variances and covariances, so it is quite complicated, and the estimates are based on the second order statistics, so it very robust and not that much reliable. So analysis is also not possible from the unreplicated data. Now, this is the difference between the Metroglyph and d-square statistics analysis. Both are genetic diversity analysis method, both used for the assessment of genetic variability, and there are some differences as well. Like in Metroglyph analysis, the statistics involved is first order, while in d-square it is second order statistics, and this is the reason how this is reliable and robust, but it is not that much reliable. And the analysis is simple in Metroglyph analysis, and d square it is quite difficult because variance covariances are involved in this. In Metroglyph analysis is possible from replicated and unreplicated data, while in d square it is possible from replicated data only. Metroglyph is a semi-graphic method, while d square is a numerical approach. And in Metroglyph analysis we are using Metroglyph chart, and or we can say that the graph, while in d-square statistics we are using cluster diagram for the identification of the genetic diversity. So this is all about the genetic diversity analysis, Metroglyph analysis and d-square statistics. Both gives information about the variability, genetic diversity, and also used for the assessment of the phenotypic variability. If you found any queries or any questions regarding this video lecture, please comment me, share, like, subscribe. Okay, thank you so much.